Thank you, Sharon, and welcome to everyone joining us today. My name is Rich Skradsky, and I'm the Northeast Regional Manager for Teledyne ISCO. Before I turn over today's webinar to Steve McManus, one of our product support specialists, I'd like to take a minute to introduce you to our company. Teledyne ISCO was originally founded in 1958 in Lincoln, Nebraska, where it has remained until this day. Our environmental division was created in 1969, specializing in the manufacture of rugged instruments used by environmental professionals in the water, stormwater, and wastewater markets. We have been protecting water quality all over the planet for the last 50 years, having invented the world's first automatic wastewater sampler in 1971. We are a true manufacturer, not just an assembly house. So when we say we are made in America, we mean it. Today, Steve will be discussing PFAS substances and how to effectively collect samples for analysis from source waters. As you can see from the map, many states have adopted or are considering adopting PFAS regulations due to the potential health impacts of this forever chemical. Recently, the U.S. House of Representatives and Senate have reintroduced clean water standards for PFAS chemicals. This would establish guidelines to help stop PFAS at the source to prevent contamination of our drinking water. We thank you for your business and stand firmly behind our pledge to help safeguard our planet's valuable water resources and to provide the industry with the knowledge to do the same. I'll now hand things off to Steve. Enjoy today's presentation.
Thank you, Rich and Sharon. Just a brief introduction of myself. Again, my name is Steve McManus. I have worked at Teledyne ISCO now for 38 years in the product support department. And as part of my role, I wear a variety of hats, from diagnosing equipment, assisting with configuration and installation assistance. I work with review of data. I work with customers in determining what products were going to be most suitable based upon their need and their application conditions. And in that vein, this is why I am here today to talk about PFAS and its role in our sampling world. Today, I'm going to have 4 basic categories that I will be covering. I will spend a little bit of time talking about what is PFAS. I will cover some sampler basics and how they apply to collecting samples for PFAS analysis. I will discuss PFAS sampling requirements, the do's and don'ts. And I will cover a little bit on PFAS analysis in the lab. So to begin, what is PFAS? PFAS is a family of chemicals, often referred to as the forever chemicals. There are perfluoroalkyl and polyfluoroalkyl substances that are categorized as PFAS. There are several related substances in this family, such as PFOA, which is often referred to when talking about Teflon. So what are the environmental concerns, and specifically the water concerns, as they relate to PFAS? Studies indicate that sustained exposure to PFAS over certain levels may result in adverse health effects. These can include, but are not limited to, the developmental effects for fetuses during pregnancy or to breastfed infants, such as low birth rate, accelerated puberty, skeletal variations. PFAS is also suspected to contribute to cancer, and specifically testicular and kidney cancers. There can be negative impacts to the liver, such as tissue damage. There can be effects on the immune system, such as inhibiting antibody production and other immunal impacts. There can be impact on thyroid glands or other effects, such as impacts to cholesterol in the body. Treatment plants, generally speaking, have not been historically tooled for PFAS removal, but that is changing. It is important to note and understand that this is a rapidly evolving science. This is one of the reasons that one researching this field can find what appears to be contradictory information. Note the dates of information that you are reviewing, as they may be out of date. As more is learned and put into practice, what was or is considered appropriate today could change tomorrow. The opposite is true as well. What was considered inappropriate today could be potentially considered okay tomorrow. So it is important to maintain contact with the most recent up-to-date information available, as it is developing at a very fast pace. This is a PFAS molecule. As you can see, it is quite a complex molecule. So what are the sources of PFAS? PFAS is everywhere. Teflon, sunscreen, shampoo, toothpaste, insect repellent, fast food containers, especially packages that have a shiny liner, chewing gum wrappers, cigarette packages, tape, gloves, clothing, especially clothing coated with water repellent and stain repellent substances, other water repellent coatings, ice substitutes such as gel packs, low density polyethylene that has not been verified to be PFAS free, firefighting foam, and a myriad of other substances. The guitar strings I use are coated with PFAS, and I use them because they last much longer than non-coated strings. I have seen TV commercials for a miracle non-stick pan that is advertised to be PFOA free. This is the reason for the high risk of contamination, because we are exposed to PFASs everywhere in our normal daily life. PFASs are all around us, and so the risk is high of carrying PFAS to your sampling site and thus cross-contaminating your collected samples. So what are the detection locations where PFAS presence is of concern? The selection of locations to monitor and sample for PFAS and detect sources contributing to PFAS is not unlike other sampling programs. Start at key junctions, and if PFAS is detected, work your way back up the watershed branches and follow the trail that has detected PFAS until the source is identified. The three main branches where PFAS detection is and will be monitored are drinking water, which is the most established monitoring and has the most defined protocols today. The wastewater sampling monitoring for PFAS is developing, and the stormwater monitoring is at the conceptual stage at this point in time. So for sampling for PFAS, the sampling basics for PFAS are not unlike other sampling purposes, but the care and approach is far more critical, like all other sampling protocols on steroids. This is due to the extreme prevalence of PFAS in our environment and the extreme small quantitative measurements of PFAS that one is looking for while sampling. Cross-contamination potential is huge, and therefore far greater care is required to avoid compromising the quality of the collected sample. The two types of sampling would be manual or grab samples versus automated or automatic sampling. For manual sample or grab sample, one can get by with a simple tool such as a pole with a bottle or a cup at the end of it that is used to dip and collect sample out of the source. So the pros are it's a very simple approach to collecting samples. The cons, on the other hand, of such a sampling routine are that one, it's less hygienic because it places personnel in the very location and risks contact with the source that we are sampling from, which could have elements that are beyond PFAS and some of which could be hazardous. It is time consuming because it requires an individual or individuals to travel to the locations to collect that sample. There could be variation in the sample collection due to different approaches that different individuals take, as well as inconsistencies in the method of sample collection that a single individual could have. It also represents a singular moment in time because it only represents the constituents in that source at the moment in time that that sample was collected. This is where automated sample comes into play. So the pros with an automatic sampler are, one, it is far more hygienic because you do not have an individual or individuals at risk of coming into contact with the source that we are sampling from. It's relatively quick and easy. You set up the sampler, you push the button to initiate the program, and you walk away. The cons of an automated sampler routine are that it does have more cost at the front end because you are purchasing sampling equipment. However, over long term, that additional cost is abated because you do not have the continual and repeated costs of having personnel travel to the location to collect those samples. So therefore, one has lowered personnel and labor costs. Again, the grab samples open the door to all the variances that can impact the integrity of a grab sample. User deviations, proper and consistent decon measures may not be followed, etc. Grab samples also increase the risk of cross-contamination due to the more direct human contact with the act of collecting that sample. And that is the main and primary benefit of an automated sampling approach, as it eliminates those variances that can impact the sample integrity. There are two basic approaches from an equipment standpoint for collecting samples. One can utilize a composite sampling approach, which is the most common means of PFAS sampling today. This involves a sampler that utilizes a single bottle, and we can collect a series of samples over a period of time, typically one day, that will be placed into that singular bottle. The samples over that collection period will be collected at a user-defined interval, most often time. Samples in that bottle represent the composite of samples collected throughout that sampling period. And again, this is the most common approach today. However, there is also the sequential or multiple bottle approach, where we can collect a single or multiple samples into a given bottle, and those samples can be collected at user-defined intervals again. And then the bottles are switched based upon typically a user-defined time interval. The example in the photo above, there are 24 bottles. So, and a typical approach would be to collect multiple samples in each bottle, and then every hour we switch to the next bottle. So at the end of the day, each bottle represents an hour of time, and we can evaluate the sample quality for the detection and presence of PFAS, and we can establish the trend of PFAS over a day, hour by hour, as a result. The pacing approaches, again, the most common one is uniform time, where we would collect a sample at every 15 minutes or every hour and place that sample into the bottle or bottles over a day. You're typically not going to be finding non-uniform time samples for PFAS sampling, but that also is an option. We can also collect samples based upon flow. If we have a flow meter in use at the location that is monitoring flow and providing flow rate, or more accurately, volume-based information to the sampler, that signal can be via 4 to 20 milliamp signal that represents a 0 to 100% flow rate. It can be a contact closure where every closure represents a fixed amount of volume. Or we can have a flow module attached to a 6712 sampler, such as we see in the photo here, that is monitoring the flow for us. And we can program the sampler to collect a sample every 200 gallons. So as flow rate increases and is more elevated, we are collecting more frequent samples. And when flow rate decreases, we are collecting less frequent samples. So the samples that are in that container or containers at the end of the sampling interval are more flow weighted in nature. Composite sampling, again, is the most common. Time-paced is also more common for PFAS sampling, but there are those who will flow pace as well. Again, assuming there is a flow meter in place to provide that flow-based information to the sampler. I will cover some materials later on in the presentation here, but at the top of the list is do not use Teflon for your intake suction line or any part of the sampler equipment. Generally speaking, high density polypropylene sample collection bottles should be used. Low density polyethylene bottles that have not been verified to be PFAS free should not be used, nor should glass. Taking equipment blanks for analysis is a frequent and standard procedure to ensure sampling components are not falsely contributing to and resulting in false elevated detection of PFAS presence in the sample. This is especially important pre-deployment of the equipment. It is recommended to draw field blanks both before and after the sample collection routine using deionized water that has been validated to be PFAS free. I have read recommendations in some of the PFAS documentation. I found that sample collection bottles should only be used once for PFAS sampling and then discarded due to the minute trace amounts we are dealing with here. For PFAS sampling requirements, there are some rigid requirements that relate to the materials that are being used in your sample program. Approved materials would be, again, high density polypropylene collection bottles, vinyl suction line that has been validated to be PFAS free, silicone pump tubing, stainless steel for components such as the intake strainer and tube couplings that are in your sample transport line, and then natural ice from verified PFAS-free water to cool the samples in the collection base. Materials that one should avoid are low density polyethylene, aka vinyl, unless it's been verified to be PFAS free. Glass containers, because glass can absorb PFAS over time and contribute to cross-contamination. However, in some areas, glass is approved for use if that glass has been tested and verified to be PFAS free before deployment. However, there is a limit on the reuse of that glass because of the propensity of glass to absorb PFAS. So you should not continue to use glass repeatedly over and over again. The bottle should be replaced. Teflon or other related materials should not be used because they are contributors to PFAS. Items or materials containing any fluoropolymer, gel packs, such as those blue ice packs you put in your cooler to keep your beer cold, should not be used because they are rich in PFAS. Gore-Tex or any other water-resistant materials, such as raincoats, boots that have had water-resistant coating applied, should not be used at the location where you're sampling. Clothing that was washed using fabric softeners should not be used on site at the sampling location. And consider any materials that both will come into direct contact with the sample, as well as those materials that won't necessarily come into direct contact with the sample, but will be present in the sampling location. Sources of contamination are very important to keep in mind when it comes to PFAS sampling. Remember, we are looking for detection in the parts per trillion range, so the potential for cross-contamination is quite significant. And this requires extreme diligence to avoid exposure to any source outside of the sample source itself that could be a carrier of PFAS. Personal sources of contamination could include shampoos, insect repellents, sunscreens, moisturizing lotion, some toothpastes, clothing, especially new clothing, will often have PFAS. Clothing that has been washed using fabric softeners or has been coated with water-resistant material. Fast food containers or packaging. Food or drink in sealed packages, such as juice boxes, for example, contain PFAS. When researching this topic, I spoke to one entity who indicated their field crew personnel are instructed not to shower or shampoo hair the day before they are deploying the equipment or going out to collect PFAS samples due to the cross-contamination potential from shampoo or soaps in general. Do not handle, consume, or otherwise interact with pre-wrapped food or snacks. Carry-out food, especially fast food or other food items, while on site during sampling. If it has a shiny paper wrapping, it likely contains PFAS. PPE could contain PFAS. However, never compromise safety for the sake of eliminating PFAS material. If there is no equivalent PFAS-free option available, personal safety equipment comes first, second, third, and onward. If faced with this issue, document the use of such and use your equipment blank sample to establish a baseline for the presence of PFAS with the equipment and the exposure potential before the equipment sampling is initiated. When in doubt, verify or outright avoid any at-risk material. Materials that are not in direct contact with the sample, but should be avoided, include but are not limited to things like GPS receivers, notebooks, clipboards, etc. They could have PFAS on their surfaces and become a source of contamination. Sampling equipment used for PFAS sampling should be dedicated to and only used for PFAS sampling. With the trace amounts of PFAS that we are looking for, we don't want to have any cross-contamination from any of the equipment used for other purposes. Decontamination is a must due to the sensitive nature and cross-contamination threat. This is often accomplished with a thorough rinse through the suction line and pump tubing with known PFAS-free water being run through the sample lines. It is recommended that you communicate with your analytical laboratory and the appropriate regulatory agency for their specific decontamination recommendations. This is because, as of today, there is no national standard established. There are guidelines, and they are under development. But as of today, there is potentially regionally specific variances that would be important to know. For sampling programming approaches, there could be a regulatory-driven element involved that dictates how samples are collected. But normal approaches for programming are acceptable. Time-is pacing samples is the most common approach, where one sample will be collected every 15 minutes over a 24-hour interval. If one is attempting to do flow pacing, a common design approach for the program is to design and program the unit such that it will collect 96 samples based upon average daily volume of flow at that site over a 24-hour interval. But ensure your configuration choices leave room for additional samples if we have an above average flow day. Note that flow-paced sampling is not suitable for applications that have a wide range in daily flow. It will be impractical and impossible to establish a flow program that will be able to handle both a very low and a very high flow day if there was a wide range of variance between those two extremes. And again, as stated initially, composite samples are the most common approach that we have for flow-based sampling, or for PFAS-based sampling. So PFAS in the lab. While this is outside of my realm of expertise, I have found these details on detection and remediation. So for analysis in the lab, there were two main approaches that I found in looking and doing research on this topic. For drinking water, using liquid chromatography and tandem mass spectrometry, EPA method 533 was a document that covered that approach to analyzing for PFAS in the lab. The other method I identified was EPA method 537.1, which is a solid phase extraction and LC/MS sampling that is able to detect PFAS down to 0.5 parts per trillion. The approaches for remediation or removal of PFAS are currently under investigation and experimentation. The problem we faced is that PFAS have extremely powerful molecular bonds that make breaking down these compounds extremely difficult. These processes are still in their early stages of development, and are continually evolving as I speak. One method is carbon filter collection followed by incineration. This has been the most common method to date, but PFAS can survive this approach at significant levels. There are approaches for chemical treatment. This again does not eliminate, but can begin to weaken the molecular bonds so that additional approaches can have greater degrees of success. For underground aquifer removal of PFAS, there has been research in using paraffin wax rods impregnated or permanganate to oxidize the PFAS and thus remove it. And then one removes the paraffin rods and replaces them with new rods. There's experimentation in using microbes that will eat the material, and this is under development. And while the lab results have had some success at this point, there's concern in the field that there would be other choices for food that these microbes will select over PFAS and divert their attention to eating and impacting those substances before going to PFAS. So again, as indicated up front, this is a fairly rapidly evolving science. And so on every one of these subjects, there is likely to be a measure of deviation over the next several months and certainly in years to come. So keep on top of this subject matter, do research, and understand where the technology and the science is taking us. And so with that, at this point in time, I would like to open the floor for any questions. I would like to thank you for taking your time today to join us for this presentation on PFAS. And with that, I will open the floor.
Well, thank you, Steve, for sharing your expertise. And I would like to welcome you to answer the questions that we have received. As a reminder to our attendees, please utilize the Q&A function within Zoom. This option allows us to pull reports after the webinar, just in case we are unable to answer your question during this airing. Thank you. And Steve, please, if you can answer our first questions.
Thank you, Sharon. I just also want to add to anybody who may have a question that pops in their head, later today, tomorrow, next week. You are welcome to submit those questions to my email address as you see on your screen, and I will be more than happy to answer them as they come in. As Sharon has indicated, we did receive a few questions, and so I'm just going to dive right on in and begin to answer as best as I can. The first question that was submitted asks, are auto samplers allowed for PFAS sampling? And the answer to that question is absolutely yes. With all of the indications of care that were given during the presentation, as long as those measures are followed and one of us avoided all of the potential sources of cross-contamination, automatic sampling is an acceptable method of PFAS sampling. And there are a number of groups throughout the country and the world for that matter that are actively using automated samplers to collect samples for PFAS analysis. Again, with the stipulation that all of the activities and concerns are addressed with the cross-contamination potential. The next question was asked, do you have any recommendations for sampling strategies? As far as specific recommendations, certainly I can make specific recommendations, but there is certainly a regional flavor to the approaches that are dictated with laboratory and regulatory agencies that may have various uniqueness compared to a general strategy that would be applied to anywhere in the world. The most common approach, again, is composite samples, single bottle, where one is collecting a sample at 15 minutes over a day. And that is the most common approach I have encountered with PFAS sampling. But again, to reiterate, there are variances that can be taken on how one approaches their sampling program, and those can vary based upon regional regulatory requirements, laboratory requirements, or even the application that one is sampling in. For example, if one would be sampling at an industrial outflow that may only have active flow between, say, the hours of 6 a.m. and 5 p.m., it would be inappropriate and a misuse of the equipment to try to sample throughout the full 24-hour interval, because for a good chunk of that 24 hours, there would be little to no flow to be able to collect samples. So there is no one-size-fits-all approach, but there are some general strategies that are consistent, and the most common one, again, is single bottle composite sampling, time-paced, 15-minute sample intervals. The next question I have here was, have you run equipment blanks on the automated samplers? Now, again here, one should take equipment blanks using the PFAS-free water, both before and even after deployment and collection of samples with a sampler, so that one has a blank baseline to compare with the active samples that were collected throughout the sampling routine. If ISCO were to have performed a test on any material that would be in contact with the sample within the factory, that would include suction line, pump tube, bottles, that would not be valid when that equipment is being deployed in the field, because between the test at ISCO and the time of deployment in the field, those materials may have come into contact with a variety of sources that could have contributed to PFAS. So the equipment blank should be performed on the equipment definitely once in the field before one activates the program, and again after. There's nothing wrong with validating using an equipment blank within your lab, your shop, to be able to evaluate the presence of PFAS before deploying to the field. But that equipment blank in the field is also critical, because again, there could have been contact with sources of PFAS between the shop and the field. ISCO is looking into providing some of that pre-analysis, but there is nothing that is going to be a substitute for that equipment blank that is taken in the field before one pushes the button to start the program. The next question regards the 24 wedge bottles for the multiple bottle sampling. Those are LDPE, low density polypropylene. So again, when using low density LDPE, one should perform an equipment blank using that material to evaluate and establish that it is PFAS free before deployment, for not only from the raw material, but for the potential for cross contamination. Again, the most common approach is not the multiple bottle, but the single bottle, which is a high density polypropylene bottle. The next question here is vinyl tubing sold by Teledyne okay for PFAS sampling? And for right now, I can say that it is being used for PFAS sampling in the field, simply because of other materials have greater risk, and there's more than just a tube involved. It's also the characteristics of the tube that can either contribute towards proper operation of the sampler or inhibit proper operation of the sampler. Tubing that obviously as Teflon or anything like that should never be considered. And other tubing materials, especially if the tubing is more softer in nature, will work against the actual operation of the pump and diminish its efficiency and its ability to reliably deliver accurate and consistent samples to the container. So there is the downside there. But for right now, there are many locations and individual groups that are out there using the ISCO samplers with the vinyl line. And as of yet, I have not encountered or heard of any problems associated with the use of that vinyl tubing. The next question here, could you repeat what the most common or recommended sample container and tubing type for use in the composite sampling? Again, the most appropriate bottle for PFAS sampling is our 2.5 gallon HDPE bottle, high density polypropylene bottle, is the most common and accepted container for use. The silicone pump tubing that is within the pump also is considered to be acceptable based upon today's standards. And then the vinyl suction line is also considered to be appropriate or has not created any issues to date. Then the other materials that would be in the sample transport would be the stainless steel tube coupler, which joins the suction line to the pump tubing, and the strainer that is in the source that you are drawing samples from. So all of those materials right now today are considered to be acceptable for PFAS sampling. There is a question here that indicates why suddenly the concern on PFAS sampling after all these years. Well, science in general is an ever-evolving situation. As we learn more, we take different approaches to our daily life. And this is no different in the world of water monitoring and water sampling. PFAS concerns are something that has been under development for a while. There certainly are challenges associated with PFAS sampling as indicated throughout the body of the presentation, because PFASs are quite prevalent in our daily life. There is very little in one's daily life where there are not multiple sources where one has and the potential to come into contact with PFAS. But the science has begun to establish that there are some risks associated with PFAS, and therefore the science of detection and sampling and monitoring for PFAS have begun to develop, and that is what has brought us to where we are today. Is there an acceptable level of PFAS or PFOA in stormwater or drinking water? The answer for drinking water obviously is yes, and as it was indicated in the presentation at the very initial stages, the most developed area of PFAS monitoring is in the drinking water. I am most involved with the wastewater and stormwater, although I do have a little bit of contact with the drinking water applications as well. I am not personally aware of any national standard for PFAS or PFOA in drinking water, There certainly are going to be regional limits and acceptable levels. In the stormwater and in the wastewater, stormwater is probably the least developed area, but in the wastewater, those levels are under development, under study, and as of today, there was no absolute acceptable levels that I have identified for PFAS in wastewater. The next question here is, do you know of any magnetized filtering option for PFAS remediation, or is this even a viable method? In my research on the remediation and removal side of this equation, I did not find anything that specifically indicated anything related to magnetization. As was indicated in the remediation section, there are a number of approaches that are under development and validation at this point in time. There is no one or two standard methods that are, being rubber stamped as the approach to take. And certainly as the science evolves, that is most likely to change. But as of right now, the bigger objective appears to be identifying where PFAS is located and at what levels. And certainly it would become quite logical that will follow will be, what do we do once we have identified and established what are the safe limits and what is going to be required to remove PFAS from the sources that we have monitored. The next question here is, there's a lot of attention around PFAS precursor compounds and their ability to form PFOA and PFOS. Can this be avoided in the sampling method? My honest answer to that at this point in time is I simply don't know. I have not encountered any information on that specific question, and therefore anything that I would say would be at best an educated guess. That is something that would probably be more suitable for a discussion with the analytical lab, and they would be able to give better guidance than anything I could provide today. The next question here is, you mentioned flushing the tube of your auto samplers with a known PFAS-free water in order to clean the equipment. Would this same approach work for collection bottles and containers for reuse? And the answer to that is potentially yes, with the stipulation that I would give. One would need to, again, take a equipment blank using, again, that PFAS-free water and collecting a sample or two into a decontaminated bottle for analysis to determine, is our approach to decontamination achieving the goal and removing and eliminating the presence of PFAS in that container. If the PFAS is beginning to show up because of use and reuse, at that point in time, the answer is to no longer use that particular container for PFAS sampling and obtain a brand new bottle to resume PFAS sampling, because again, we are looking for the presence of PFAS in such minute quantities. We do not want to have a false detection of PFAS be the result of a sample bottle that has been used and reused multiple times in a given application and has begun to retain PFAS. Okay, the question may be out in left field. However, are you aware of any restrictions on internal fire suppression systems using PFAS, such as in airport hangars where most systems use water? Well, in many cases, especially when you're dealing with airports, fire suppression foam is highly containing with PFAS. So this becomes the case of where is the greater harm or the greater good. If you have a fire that has begun to begin for whatever purposes, and you have fire suppression foam that is PFAS, what is the greater good? Putting out that fire right now, or not using the fire suppression foam because of the presence of PFAS and allowing that fire to grow and potentially come out of control? Seems to be a fairly logical answer to me. However, that certainly could change with time. Maybe additional substances will be identified over the next several months, years to come that are PFAS free for being able to achieve the job of fire suppression. And certainly when that comes into play, the likelihood of elimination or removal of today's methodology with fire suppression foam that is PFAS will begin to be removed from the equation. But as of right now today, they're just simply because of the availability of what materials are available to achieve the goal of, well, putting out the fire, having PFAS, and alternatives are far and few between. Today, there is no restrictions or regulatory changes in that approach. The next question is sample preservation required? And what is your recommendation for sample storage and holding time? Well, here's what I'm going to say on that. First of all, one should always communicate with and understand what the analytical laboratory's requirements are. That's going to supersede anything that I will say right now today. However, there were in a number of the documents that I reviewed in preparing and studying for this presentation today that did indicate the use of PFAS-free, verified PFAS-free ice for sample preservation. So I would say, in generally speaking, yes, there is preservation required, and the methodology for preservation was PFAS-free ice. But I again will reiterate and stress, communicate with your analytical laboratory and get their recommendations for what they require for that sample preservation. That's going to be far more reliable than anything I have to say today. Do I have a list of references for studies using automated samplers specifically for stormwater? I have done literature searches, but I haven't turned up much yet. I would say that's the same result that I encountered when researching this. My primary focus in my studies is I was looking for the information in detail that related to all of the above. The drinking water analysis is typically a slightly different approach, slightly different equipment. Our samplers, our automated samplers, are primarily used in open channels. You do not see too many open channels when it comes to drinking water. Most of the drinking water is closed pipe, often pressurized pipe applications, and therefore the activity of collecting samples for analysis is going to utilize different equipment for that type of application, although there are means to be able to use ISCO samplers in pressurized closed pipe applications. So therefore, the bulk of the detail that I was focused on was for the water, wastewater, and the stormwater applications. The wastewater is the most actively developing sampling routine or application at this point in time. Stormwater is in the beginning of stages for its sampling routine. So therefore, there was not a whole lot of detail that I found that focused on stormwater sampling. The bulk of what I was able to identify was for the wastewater application. However, again, as indicated multiple times over and over, the applications for PFAS sampling are a very rapidly developing science. So things are on a constant state of flux and changing as things are learned and determined based upon what is happening today. So the advice I would have here is to continue to stay on top of details, research and try to identify what the most recent application requirements and approaches are, because they will change. And therefore, what happens next month, next year, as things develop and become more refined, you're staying on top of the approaches that are necessary for PFAS sampling. Are you saying, the next question here, are you saying that HDPE, high density polyethylene bottles, or high density polypropylene bottles? It's LDPP, low density polypropylene, and high density polyethylene. The high density polyethylene is, at least as far as the sample bottles are concerned, we have for our sample bottles, for the multiple composite sampling bottles, we use the 24 and these are the wedge shaped bottles, the low density polyethylene. Now, low density polyethylene, generally speaking, has not been the primary material for sample collection bottle, but low density polyethylene, if it's been verified to be PFAS free, can be used. The single composite bottles we provide are HDPE, or high density polyethylene, and those are PFAS free. But again, as indicated within the presentation, as a bottle is used and reused, the potential is there for that bottle to begin to collect and begin to contribute PFAS to your collected sample. So this is where the equipment blanks are very important to establish that your bottle is still PFAS free and not at risk of impacting your sample analysis. The next question here is that, do sample preservation and holding times vary upon the particular PFAS substances being collected? I did not find or locate anything in the documentation that was more specifically focused on a given type of PFAS. It was more general in nature. However, again, here I'm going to fall back on my recommendation when it comes to preservation methods and holding times. Communicate with your analytical laboratory and get their recommendations. That is the most reliable and accurate source for any information related to those particular